Hello everyone, and welcome to our channel, MooTechie. This presentation is an introduction to enzymes and their functions. Looking at the slide, we also see a reference to the catalytic center of carbonic anhydrase, where a histidine triad chelates a zinc ion to form the catalytic site. There's also a mention of a small molecule that mimics this enzyme, called a tri-Beck chelate, with work attributed to Lawrence Livermore National Laboratory and a researcher named Single Wang. But our core focus begins with the basics. Enzymes are biological catalysts. They speed up reactions by lowering the activation energy, stabilize the transition state without being consumed, and have a high specificity for their substrate and reaction. Now let's look at enzyme structure and the active site. The 3D fold of an enzyme forms an active site pocket. The substrate binds via precise non-covalent interactions. Specificity comes from the shape and the side chain chemistry of this pocket. Some enzymes require cofactors or coenzymes for their activity. The slide shows a diagram illustrating this process, a substrate entering the active site of an enzyme, forming an enzyme-substrate complex, then an enzyme products complex, and finally the products leaving. The enzyme changes shape slightly as the substrate binds. Next, we explore activation energy and biochemical reactions. All reactions must cross an energy barrier. Enzymes provide a lower energy pathway. The overall change in free energy between reactants and products remains unchanged. Only the rate increases. The slide contains a graph with two curves. The higher curve shows the activation energy needed without an enzyme, while the lower curve shows the reduced activation energy pathway when an enzyme is present. The axes are labeled energy on the vertical and reaction progress on the horizontal, going from reactants to products. Coming up, we'll discuss the transition state theory for enzyme catalysis. The rate of a reaction depends on forming the transition state. Enzymes bind and stabilize this transition state more tightly than the substrate itself. This stabilization dramatically lowers the activation energy. Because of this, transition state analogs are often potent inhibitors and useful scientific probes. The diagram on the slide shows the reaction pathway comparing the higher energy transition state of an uncatalyzed reaction to the lower energy transition state when an enzyme binds and stabilizes it. Following this, you'll notice the first of two classic models of enzyme action, the locking key model. This suggests a rigid complementarity like a lock and key, where a pre-shaped active site fits the substrate perfectly. This model explains specificity well, but is less effective at explaining catalysis. A simple visual on the slide shows a fixed active site and a matching substrate coming together. The second model is the induced-fit model. Here, binding triggers a conformational change in the enzyme. The active site molds around the substrate, which aligns catalytic residues and optimizes transition state stabilization. This model better accounts for enzyme promiscuity and regulation. The slide has a diagram showing a flexible enzyme, colored in blue, changing its shape to accommodate an orange substrate, leading to optimized catalysis. As we continue, let's understand the general mechanism of enzyme action. The basic steps are numbered on the slide. 1. Substrate binding. 2. ES complex formation. 3. Transition state. And 4. Product release. The overall cycle is often shown as E plus S in equilibrium with ES, then ES to EP, and finally EP to E plus P. Modes of catalysis include covalent, acid-base, approximation and orientation, and metal-ion catalysis. Some enzymes also show cooperativity between different states.
Moving forward, we'll see how enzymes are affected by their environment. Starting with temperature, the reaction rate rises with temperature up to an optimal point. High temperatures disrupt the enzyme structure, causing denaturation. This optimum reflects the organism's environment, for example, around 37 degrees Celsius for human enzymes. A chart on the slide shows a bell-shaped curve of enzyme activity percentage versus temperature. Similarly, enzymes are affected by pH. Activity peaks at an optimal pH because pH alters the ionization state of critical active site residues. Extreme pH levels cause a loss of structure and function. The slide features a chart showing a curved peak of enzyme activity percentage across a pH scale from 0 to 14. Now, let's look at substrate concentration. The reaction velocity increases with substrate concentration until it reaches a maximum velocity, called Vmax, when the enzyme becomes saturated. The Michaelis constant, Km, gives an apparent measure of the enzyme's affinity for its substrate. At low substrate concentration, the rate is nearly proportional to it. At high concentration, the rate approaches Vmax. This relationship is shown in the classic Michaelis-Menten curve on the slide, plotting reaction velocity against substrate concentration. Next, we explore enzyme inhibitors. There are three main mechanisms illustrated with simple diagrams. First, competitive inhibition, where the inhibitor binds directly to the active site, competing with the substrate. This increases the apparent Km while Vmax remains unchanged. Second, non-competitive inhibition, where the inhibitor binds to an allosteric site, changing the enzyme shape. This decreases Vmax while Km remains the same. Third, uncompetitive inhibition, where the inhibitor binds only to the enzyme substrate complex. This decreases both Vmax and Km. Now, let us look at apoenzymes and holoenzymes. An apoenzyme is the inactive protein portion. To become active, it often needs helpers called cofactors, which are inorganic ions like magnesium or zinc, or coenzymes, which are organic molecules like NAD+ or CoA. When an apoenzyme binds its necessary cofactor or coenzyme, it becomes a complete active holoenzyme. These helpers enable crucial functions like electron transfer or chemical group transfers. The slide has an image showing an inactive apoenzyme combining with a metal ion and a coenzyme to form the active holoenzyme. Thank you for joining this lesson on enzymes. We hope you enjoyed learning about these amazing biological catalysts. Please subscribe to MooTechie, like and share if you found this helpful. Share with your friends, and feel free to comment with your questions. Thank you. See you in the next class.